right, everybody, welcome to the lecture, the pre-recorded lecture for chapter 27, Bacteria and Archaea, the two excuse me, the two domains that we don't talk about as much as we probably should. If for no other reason than these organisms represent perhaps some of the clues or the keys to how life might have actually, once it began on Earth, which was the subject that we talked about back in chapter 25, how it might have proliferated after that. And the reason why I say that, the reason why I say that it's this group that we generally credit was being the first critters on the planet has a lot to do with the fact that they live in all kinds of environments. They're what I call extremophiles, okay? Organisms that love, or at least some species anyway, that love extreme conditions. They can live where it's really warm, over 80 degrees Celsius, right? And as I see in the thing, there's the ones that live near those geothermal vents, like out in Yellowstone. The water there is very close to boiling. Remember, boiling is 100 in Celsius, so living in water over 80 degrees Celsius is pretty hot water. So that's why we call them, I'm writing the word out, extremophiles, organisms that love or can deal with extreme environments. Now, the Archaea domain, the Archaea, that actually comes from a word archaic, which means very old. So Archaeans are some of the older and the more extreme aphilic type organisms out there. Archaeans can live in just incredibly bizarre environments. Some prokaryotes, so we lump these both together, of course, by cell types, we can call them prokaryotes. Some prokaryotes in these two domains can withstand radiation doses well beyond what would kill us. 3 million rads, which is 3,000 times the fatal dose. So if you get popped with 3 million, whatever, oh man. If you get hit with a fatal human dose of rads, whatever that might be, what we mean by that is, of course, it's going to kill you. I'm trying to figure out what that would be with 3 million. It's 3,000 times more. But the math is unimportant. That's an awful lot of radiation that these organisms can deal with and survive, which means perhaps that the whole idea of panspermia might even be expanded into not just amino acids being spread throughout the environment. There could have been perhaps relatively primitive, no, not primitive, let's call them relatively ancestral type organisms, right? Ones that can deal with this kind of environment where the radiation, remember out in space, thank goodness for our magnetic field on Earth. That's what keeps a lot of the radiation off of us. But out in space, you don't have that. And radiation is a quick killer. A lot of these organisms can live in environments that you can only possibly imagine. Like, for example, pH is down close to 0. And remember, in the pH scale, the further you get away from 7 on either side of the scale makes you an acid or a stronger acid or bases. So acids have pH less than 7. So a pH of 0.03 is extremely acidic, enough to, like, it's kind of like the blood in Alien. Remember the creature? They cut the appendage and that blood just eats through the metal. Same thing. This stuff will dissolve metal. And these organisms can live in it, right? Or salinities that are just outrageous. Salinities, you think about the osmotic problems associated with living in salinities greater than 27%. Now, the ocean, just as comparison, the ocean, and think how salty that is only 3.5%. So that's somewhere in the vicinity of 8 to 9 times more salty than the ocean. So from an organismal point of view, from a biologist point of view, it would be interesting to say, gosh, how the heck do they deal with that? How do they deal? Radiation causes genetic disorders. It knocks off those bases, A's and G's, and messes them all up. How do these critters deal with that? How do they deal with those pHs, the temperatures, and the salinity? How do they deal with such a hypertonic environment? And not only do they deal in these, live with these really radical environments, they are everywhere. If you took, my Bio 1 class knows this. If you take all the prokaryotes on Earth and you put them into one side of a scale, a balance, so over here is all the prokaryotes, all of them, right? Every bacteria, every archaean on Earth, everything. And on the other side, you put all the eukaryotic organisms, which hopefully you remember from Bio 1, the plants, the animals, the fungus, and the protozoans, that weight, not numbers, folks, just weight, mass, just pure mass, would tip the scale towards the prokaryotes. They are everywhere. As you can see in this picture, this is one of those electron microscope pictures. of one of those, of a common ordinary object. I love those kind of photographs where they take something and they magnify it a bunch of times, and then they go, oh, this is the wing of a butterfly. And you go, oh, that's so cool. I didn't know that. This, if you the electron microscope picture, happens to be the head of a straight pin. You know, a regular old sewing straight pin. And all the little yellow things that are growing on there are bacteria, specifically rod-shaped bacteria. You can see why they're called rod-shaped, because, excuse me, they're shaped like a rod. Now, the colors are artificial. My guess is they may have stained these bacteria, but look how many there are. So you think about that the next time you go and use a straight pin to dig a syringe, to dig a splinter out of your finger, right? If you go on and you take A&P, or if you've ever had any A&P types courses, you know that our number one defense against disease is an intact skin or an intact integument. So when you poke a hole in there with that straight pin, you compromise your number one defense against disease, and you then also what we would say inoculate or introduce all those bacteria into your body. Now, before we get too much further into this prokaryotic chapter, all right, and I'm sorry, I'm going to use the word bacteria to refer to archaeans and bacteria many times. So I'm going to switch back and forth. So if you get confused, make sure you let me know. So I like to collectively, let's just call them prokaryotes. While the prokaryotes are everywhere, while there's more of them weight-wise than there are the rest of us, you know, and while you do have an immune system that fights against that, keep in mind that let's not get too crazy germophobic. Most prokaryotes do their thing. Most prokaryotes live their lives as prokaryotes doing their prokaryotic thing, and they don't bother with us. They don't interact with us. I mean, some interact with us even in a positive way, right? Like cheese, yogurt, those are all things that are made through bacterial cultures. But of course, the ones we normally hear about are the ones that cause disease. So keep in mind that while the world is covered in these prokaryotic critters, there are 10 times as many of these types of cells on and in your body than your entire body is made up of. Human bodies are, I don't know, trillion cells, I think they say. Living on or in our bodies are somewhere in the vicinity of 10 trillion prokaryotic organisms or cells. Remember, because they're single-celled. So we don't want to get too freaked out about this. Remember, like I said, it's the bacteria that we hear, or the prokaryotes that we hear about, that we think are the ones that cause disease are the ones we typically hear about. There are many out there. Now, back to the panspermia for a second. This is an interesting photograph back in the late 90s. Can't remember exactly when. We found a meteorite in Antarctica. Now, a meteorite is a meteor that hit the ground, right? There's nothing different. It's just a big rock came from space. Now, for reasons that involve geological analysis, we know that this meteorite originated in Mars, or on Mars. So what happened is, we think, is Mars got hit by something, asteroid or meteor, you know, millions of years ago. Chunks of Mars got thrown up into the air, you know, like an impact would do. A lot of that stuff fell back onto the planet because of gravity, but some of it got thrown up far enough away that it actually went off into sort of interstellar space and floated for millions of years, let's say, until it got close enough to Earth, and then there's gravity sucked that rock in. Well, this rock, like I said, for geological evidence, we have very strong evidence to suggest it's originally a Martian rock. Had this had electron microscope picture, had this structure in it. And the structure I'm looking at is probably the one you can see, the one right in the middle. Almost looks like a worm. Now, it's not a worm, and this is at a very, very, very small microscopic level, but it's shape-wise anyway, it's not too unlike those rod-shaped bacteria that you see in that picture, right? Now, the question is, of course, is this structure we see just something that by coincidence that was formed geologically? In other words, is it not something to do with life, or is it truly a fossil remain of a prokaryotic organism? Remember, we talked about fossils, and with prokaryotes, they don't fossilize very well, considered, you know, really hard structures. Now, to be fair, there are geologists out there that suggest this could be a geological feature. There are astrobiologists, of course, that have reasons to suggest that it might be actually a fossilized bacterium. So in essence, or I guess in end, the jury's still out, right? We never really prove anything in science. One of the things that would help support the theory that this was indeed a fossilized Martian prokaryote would be to find some living Martian prokaryotes, right? Which would be something very exciting if we were to do that. But of course, this kind of, again, flirts with that idea that we talked about in chapter 25 about where life on Earth began. Maybe life did not begin on Earth. Maybe life just arrived on Earth, right? Okay, so let's look at some big features that we know about prokaryotes. Again, and in very broad brush, because of course we're talking about two big domains here. And just for a second, I want you to appreciate what that means. When I was in college, we didn't have domains. It started at kingdom. And there was one kingdom for all of the prokaryotes, Kingdom Monera. What we have found in subsequent years, mostly through DNA analysis, of course, is there's as much difference between a prokaryotic bacteria and a prokaryotic archaean as there is between those organisms and all the rest of us in the domain eukarya. So we have elevated those into their own separate domains. So we have two prokaryotic domains. But again, appreciate the fact that even though to us those look like two, it's back to that species thing again. Even though those two critters are small, rod-shaped, microscopic, and they kind of all look alike, right? I mean, in that picture of the needle, there could probably be some bacteria and some archaeans mixed in there. I don't think so as far as the archaeans, but nonetheless, to us, they kind of look alike. But we have learned, as I said, and that's why they're in two different domains now, that there are significant difference between those two organisms. So again, don't get fooled by that just because how something looks tells you a lot about how complex it is or how related it is. But no doubt, yes, most prokaryotes are unicellular. I didn't say all. I said most are unicellular. Unicellularity was the beginning of organisms on Earth. Organisms on Earth, or life on Earth, began as a unicellular critter. Now, later on, for reasons that I will kind of elaborate on as we get into, especially when we get into animals, we'll talk about the advantage or the evolutionary significance of cells binding together. In other words, cells creating tissues, if you remember from Bio 1. But I want to caution you that it isn't a line between being unicellular and being a tissue. In other words, just because you're multicellular doesn't mean you're a tissue. A tissue is a group of cells that performs a specialized function. You got nervous tissue, digestive tissue, pancreatic tissue, all kinds of tissue. And they form, as I said, they form a collection of cells that does something specialized. So while you might have many cells, like for example in prokaryotes, some species do gather together, and sometimes for a little bit longer times than others, sometimes temporarily, sometimes a little bit longer, sometimes in response to the environment, oftentimes in response to the environment. And while we might think of those as being multicellular, the better word for them is actually colonies. They're not quite multicellular yet. You wouldn't call all the cells in your body a colony of cells, would you? Right? That's a collection of cells. You wouldn't call all the collection of cells in your nervous tissue. You wouldn't call it a colony of neurons. That's nervous tissue, right? So think about a colony as being a group of cells living together, maybe like a subdivision, right? A group of folks living together, but they're not family, right? They don't have a common functionality, if you will. Although, and remember, we spent an entire month on evolution, and hopefully we're going to start, begin to understand why that is. And you remember why we spent so much time on evolution is because changes that occur in organisms don't necessarily appear overnight, even if it looks like in the fossil record they did, right? That was the Cambrian explosion problem. But remember that just like we finished up talking about where eyes may have come from and all that sort of stuff, we can even see in these organisms that aren't truly multicellular, and actually multicellular is even a little bit different than tissues, because sponges, which are animals, are multicellular, but they don't have tissues. But even amongst these groups of cells, these colonies, we do see a division of labor. So we do see a specialization beginning to occur. Does not make it a tissue, right? But you can see where the roots for that are laid in the foundation of what we know were early life forms. In other words, these organisms are the descendants of early life forms. So the way they lead their lives, remember in evolution, there's no goal. Just because the earth has changed, as long as whatever environment you're in hasn't changed from the way it was in the early earth. Say if you lived around one of those geothermal vents, right? It hadn't changed a whole lot. You sometimes we call them living fossils, organisms that are basically alive today that have not changed very much at all. A better example, well, not a better example, but a really good example here in Florida is the horseshoe crabs. Horseshoe crabs are living fossils. What we mean by that is, if you look at them, they look kind of bizarre, don't they? They look like ancient critters that you might see in some of the pictures from what the ancient seas used to look like. But horseshoe crabs have been around pretty much exactly the way you see them today since the time of the dinosaurs, 65 million years or so. All right. So you understand what I'm saying about this? So when you see things like colony, unicellular, multicellular, division of labor, take all that kind of with a grain of salt. Realize that it isn't really technically defined a tissue until we get to animals, although that term is used in other organisms as well. But we'll talk a lot more about that as we get into those organisms. So sometimes they bond together. Mostly they're unicellular. They got basically three shapes: round, rod, or helical. A helix is DNA is a double helix. Helix is a spring, a winding thing. So we have the spherical ones are called cocci. I always remember that easy enough because there's an O in cocci, which is round. And they can occur as pairs, diplococci. So two round ones together would be diplococci. They can occur as a chain of round ones and be streptococci. I remember that because it reminds me of a strap. And then the staphlo, the Y, reminds me that it looks like a group of grapes. All right, now this becomes very useful for folks who study prokaryotes because shape and the way they arrange themselves as twos and chains and grapes can help in identifying these organisms. I know you think, wait a minute, I thought you told us not to use morphology. No, I said you can't use morphology as the defining characteristic, but it is a good place to start. And if you take microbiology, one of the things here at Lake Sumter, one of the things that Professor Wood or Professor Otis will have you do is be able to identify prokaryotic organisms. And one of the ways you're going to start, again, is with shape. All right, so cocci if they're spheres, bacilli if they're rods, spiral, or sometimes that last one is called spirillum, which means helical shaped, okay? And of course, they're small, right? We talked about that at the beginning as to why that was kind of misleading when you think about them all being lumped together in that old kingdom that I learned, the kingdom Monera, right? Being small, it doesn't have anything necessarily to do with you being related to one another for the same reasons as, having flippers if you're a dolphin versus having fins if you're a fish means that you're closely related. Remember, that's all a relative term, closely related. Some prokaryotes get pretty good size. Most are about 1 to 5 microns, right? It's all about that surface area to volume ratio from Bio 1, right? Cells stay small in general, even if it's a eukaryotic cell, which can be, 10 to 20 times bigger. Generally, cells are going to stay small. This one here, Thiomargarita nambiensis, I think it's because it was described from Nambia in Africa. Thio is a sulfur. So Thiomargarita is a sulfur bacteria. It's a bacteria that actually makes its living off of oxidizing sulfur, right? If you're wondering what the heck of that bee is doing there, they're trying to give you a little comparison. Here is Thiomargarita nambiensis, which is about pretty good size for a single cell anyway. On it, there it is right there on the bee. So you can see it. You can see it with the naked eye. But that's unusual, right? Most of the time they're a lot smaller. Okay. They do have a cell wall. They also have modifications to their cell surfaces, but first of all, let me clarify the cell wall thing. It's not the same kind of cell wall you see in plant cells. It's not made of cellulose, but, and here's an example of convergent evolution right down there at the cellular level, right? This cell wall, and we'll talk about some of the modifications to surface structure too, but the cell wall does, in the functions down below, something that you probably talked about when you talked about the cell wall in plants, right? It maintains the shape of the cell. It protects. It's an outer armor type coating over the cell. And remember, plant cells you can also use that cell wall to prevent themselves from lysing, right? Plants are all about the water. They got to get water, and they can't go get it, and they got to store it. So having that huge central vacuole filling up with water would be a problem if you didn't have a cell wall, right? It's form and function, right? Think about that when you're trying to figure out, okay, how does this all fit together? Who's got what? Now, as I said, prokaryotes have a cell wall too. While it does the same functions, though, it has a very different structure. So again, convergent evolution, right? Right at the cellular level. So there's nothing magical about convergent evolution other than organisms are finding ways to adapt to similar habitats. It doesn't matter whether you're a plant cell. It doesn't matter whether you're a prokaryotic cell. If you live in a hypotonic environment, you're going to take in water. That's a law of physics and chemistry, and those laws were around before there were living organisms. Hopefully, one of the things you took out of Bio 1, like especially with respiration and photosynthesis, is what living things do is they hijack, in a way, the natural tendencies for things to diffuse, and they use that to do work. So we're going to find similar solutions. So convergent evolution is not something rare, right? It's actually pretty common. Okay, but let's look at, there's the similarities. Let's look at the differences between the cell walls we find in prokaryotic cells and the ones we saw in plants. And I'm not going to show you the plant ones because, that's the cellulose fibers. All right, now, there are two types of bacteria, domain bacteria organisms out there. And they're called gram-positive or gram-negative. Now, we'll talk in just a second about how you determine whether they're gram-positive or gram-negative. But the name comes from a type of stain, okay? If an organism, if a bacterium is gram-positive, when you stain it, it will look purple. And if an organism is gram-negative, when you stain it, it'll look reddish. You can see down below, I've labeled a couple of them, right? You see they're very simple. Gram-positive purple, right? Gram-negative. Now, this is, again, another way to help identify organisms. So now you could say, what shape does it have? And gram-positive and gram-negative breaks up bacteria into two big groups. It'd be like saying bird or fish, right? Now, let's talk a little bit about that cell wall, because the basis of whether these things are gram-positive or gram-negative does have something to do with structure again. Now, here's the cell wall, not made of cellulose, but made of this stuff called peptidoglycan. All right, it's a polymer of many materials stuck together. Notice that there is a plasma membrane with those embedded proteins, just like you learned about in Bio 1, but on the outside of that is this peptidoglycan layer, okay? Now, the gram-positive bacteria have the cell membrane and the peptidoglycan, end of story. Gram-negative are a little bit more complicated. They actually have